What if, instead of just treating diseases, we could program our own cells to become microscopic doctors? What if we could task them with hunting down and eliminating threats from inside our own bodies? This might sound like science fiction, but it's the basic idea behind a field of research that's happening right now. We're on the edge of a revolution where medicine can start to make its own decisions. For most of modern medical history, we've relied on static things: pills, injections, infusions. It's often a one-size-fits-all approach. A drug goes in, spreads throughout your entire system, and often causes collateral damage to healthy cells. The problem is that a single pill or shot can't tell the difference between a sick cell and a healthy one. The body's complexity really demands a smarter, more targeted solution. But what if the medicine itself was alive? That's the promise of Engineered Living Therapeutics, or ELTs. These aren't just chemicals, they're living cells, often common bacteria, that have been genetically programmed to act like those microscopic doctors. Unlike a normal drug, these living therapies can sense what's happening around them, react to signs of disease, and carry out complex therapeutic jobs in real time. It's a huge leap from medicine as a passive substance to medicine as a living, responsive system. And the wild part is, this is no longer just a theory. Some of these microbial therapies have already been advancing into clinical trials for treating cancers and metabolic disorders. So, how close are we, really, to using programmed bacteria as medicine? Let's take a look. The big idea, core concept, the core idea behind Engineered Living Therapeutics sounds simple, even if the biology is incredibly complex. You start with a living cell, often a well-understood and safe bacterium like E. coli Nissle, and you give it a new job. This reprogramming is done using the tools of synthetic biology. Scientists insert new pieces of DNA into the cell, creating what are called synthetic gene circuits. You can think of these circuits like the logic gates in a computer. They can be designed to function like literal Boolean operators, or not gates, but they're built with the language of DNA. These circuits allow the cell to process information from its environment and make pre-programmed decisions. For example, imagine a bacterium programmed with an AND gate. It might be engineered to only release a cancer-killing drug when it detects both a specific tumor marker and the low oxygen environment typical of a solid tumor. This keeps the therapy from activating anywhere else in the body, which could dramatically increase its precision. A NOT gate could be used to shut down the therapeutic function if the cell senses a molecule that's only present in healthy tissue. By combining biology, engineering, and computer science, researchers can now design and build these biological systems. A term you might hear from regulators like the FDA is live biotherapeutic product, or LBP. This is a category for a biological product that contains live organisms, like bacteria, and is used to prevent, treat, or cure a disease. And while we're all familiar with probiotics that support gut health, these engineered therapeutics are a completely different beast. An engineered bacterium is designed for a highly specific mission: sense a disease biomarker, produce a potent drug molecule exactly where it's needed, or selectively get rid of harmful microbes. That's the big difference between a passive pill and a dynamic, living therapeutic. It can, in theory, live in the body, adapt to changing conditions, and deliver treatment only when and where it's required. Once you can reliably program a cell, you're not just making a medicine. You're designing a living machine. Deep dive analysis. So why is this such a big deal? To get it, you have to think about the fundamental weaknesses of traditional drugs. Many therapies fail because they break down before reaching their target, they can't be concentrated enough at the site of the disease without becoming toxic, or they require constant, repeated dosing. Living therapeutics are being designed to solve these exact problems. Their biggest advantage is what's called in situ manufacturing, making the therapy directly inside the body, right at the spot where it's needed. For example, researchers have engineered strains of E. coli that are being investigated for their potential to produce anti-inflammatory compounds on the spot when they sense inflammation in the gut. This could offer a focused therapy for conditions like inflammatory bowel disease, IBD, with fewer systemic side effects, although so far, these have shown promise in preclinical models but haven't yet demonstrated clear efficacy in large human trials. This is all made possible by what researchers call sense and respond systems. The engineered cell has a sensor, its eyes, that can detect disease-related molecules, like lactate in a tumor or specific inflammatory signals. This input then triggers a pre-programmed response from the cell's genetic logic, its brain. The response, or payload, could be the release of an anti-inflammatory protein, an enzyme that breaks down a toxic substance, or a powerful molecule that calls in the body's own immune system to attack a tumor. In some studies, scientists have engineered bacteria that are naturally drawn to the low oxygen regions inside tumors. While not quite a tumor GPS, this natural tendency can be enhanced with further engineering to help the bacteria accumulate where they're needed most. Once there, they can be programmed to release molecules that stimulate the immune system, potentially making other treatments like immunotherapy more effective. But of course, this leads to the most important question of all: is it safe? If you're putting a live, engineered organism into a person, you need absolute confidence that it will behave predictably. This is why biosafety is one of the biggest focuses of the entire field. Many designs now include multiple safety features. Kill switches are genetic circuits that cause the bacteria to self-destruct on command, for instance, when triggered by an external drug. Containment strategies are also being developed, like engineering bacteria that depend on a nutrient that isn't available in the human body, so they can't survive for long. However, making these safety switches truly foolproof remains a challenge, as microbes can sometimes evolve to escape these controls. In a novel approach, researchers at Harvard's Wyss Institute developed implantable, living materials by encapsulating engineered bacteria inside a specialized hydrogel. This keeps the bacteria physically contained at a specific site while still allowing them to release therapeutic molecules. These strategies are crucial, because the goal isn't just to prove the biology can work, but to build systems that regulators, doctors, and patients can trust. If you're fascinated by science that sounds like science fiction but is actually happening right now, this is exactly the kind of topic this channel is all about. So if you're getting value from this, please take a moment to like the video and subscribe. It really helps the channel grow, and it tells the algorithm that more people need to know about the future of medicine. Real-world and human impact. So where will we see the first real-world impacts of this technology? The most immediate applications are being tested in diseases affecting hard-to-reach or complex environments, like the gut, solid tumors, and chronic wounds. For inflammatory bowel disease and other gut disorders, engineered probiotics are a natural fit. Companies have also pushed forward with clinical trials for engineered bacteria designed to treat metabolic disorders by consuming toxic substances in the gut. For example, the company Synlogic ran trials for phenylketonuria, PKU, a rare genetic disorder, with a synthetic biotic designed to break down the harmful amino acid phenylalanine. While early results were promising, their main PKU drug unfortunately failed its final phase of clinical trials in early 2024, and the program was shut down. It's a tough reminder that this is still an early and challenging field, where promising science doesn't always translate to clinical success. In cancer, the potential is enormous. The success of CAR T-cell therapy, where a patient's own immune cells are engineered to fight cancer, has already proven that programmable cells can be a powerful medicine. Now, researchers are applying similar logic to bacteria. Because certain bacteria can preferentially accumulate in the low oxygen, chaotic environment inside solid tumors, they are being engineered to act as Trojan horses. Some are programmed to deliver immune-stimulating molecules to wake up the immune system and help it recognize the cancer. Others are designed to produce enzymes that convert a harmless substance into a potent chemotherapy agent, activating it only inside the tumor and sparing healthy tissue. And the applications go beyond just releasing drugs. Researchers are creating living diagnostics. Imagine engineered bacteria in your gut that could detect the earliest molecular signs of colon cancer and produce a harmless, colored signal in your stool. One team has already developed a paper-based diagnostic platform using freeze-dried, cell-free components from engineered cells that can detect the Zika virus, showing how this technology could be made cheap and accessible. But perhaps the most profound shift is how this could change treatment itself. Instead of needing repeated hospital visits for infusions, a patient might one day receive a living implant, a small, contained device with engineered cells that provides a continuous, adaptive therapy for months or even years. We are moving from medicine as an event to medicine as a continuous, responsive system. Future implications and AMP challenges. Looking forward, the future of this field won't just be about engineered bacteria. It will be about smart, hybrid systems that combine biology with other cutting-edge technologies. Think of engineered microbes controlled by AI algorithms to optimize their therapeutic output, or living cells paired with wearable sensors that monitor your body and adjust the cell's function in real time. We are talking about medicine that is not just personalized, but truly adaptive. The possibilities could go beyond treatment and into prevention and enhancement. Imagine synthetic vaccines that use engineered genetic sequences to create a faster and safer immune response. Or engineered microbes that could be used for bioremediation to clean up pollutants. The ability to program biology has the potential to solve problems far beyond medicine. However, we have to be realistic about the very real barriers that remain. Genetic stability is a major challenge. If the engineered cells mutate, their programmed behavior could change in unpredictable ways. Manufacturing these living drugs is far more complex than making chemical pills. Ensuring strain stability, shelf life, and consistency at scale is a massive undertaking. And then there's the regulatory pathway. A living, responsive medicine doesn't fit neatly into the old categories of drug or device, and agencies like the FDA are working to adapt their existing frameworks for biologics and cell therapies to evaluate them safely and effectively. Finally, public perception is critical. The idea of putting genetically engineered bacteria inside your body is, understandably, a lot to process. The solution isn't hype. It's building trust through transparent testing, rigorous safety engineering, and honest communication about what these systems can and can't do. So, what should you take away from all this? First, engineered living therapeutics are not a magic bullet. They won't replace every pill. But they are poised to be incredibly powerful where conventional medicine struggles, in chronic diseases that require adaptive dosing, in hard-to-target areas like solid tumors, and in localizing therapy to minimize side effects. Second, this is one of the most powerful examples of synthetic biology changing the world. For the first time, we are moving beyond just discovering natural molecules to treat disease. We are learning to write the code of life itself, to program cells to behave in the ways we want. And third, the ethical and safety questions are just as important as the science. The success of this field will depend not just on whether the biology works, but on whether we can build systems that society can trust. We are moving from medicine that acts on biology to medicine that is biology. The next generation of medicine might not be a pill you swallow. It might be a living system that you host, a system that knows what to do because we programmed it to understand the body itself. So, here's the question I want to leave you with. Would you trust a living, programmed medicine inside your body if it meant a smarter, more effective treatment? Let me know your thoughts in the comments below. And if you want more deep dives into the strange, powerful, and very real future of science and technology, make sure to hit like, subscribe, and I'll see you in the next one.